This is the lecture for Chapter 13, Viruses and Prions, Part 1. I've already discussed the prokaryotic microbes, the eukaryotic microbes, and this chapter goes over the last two groups of microbes, the viruses and the prions. And I want to begin with some general characteristics of viruses. First, viruses are very, very small. They are so small we of course cannot see them with the naked eye or even a light microscope. In order to visualize a virus, you need an electron microscope. And electron microscopes weren't developed until about the 1930s, so we were unable to see viruses until about that time. Another thing about viruses is that they are not cells. They do not have cellular structure, so they do not have a cell membrane that is a selectively permeable barrier. Instead, viruses have a protein coat which protects some sort of genetic material. And that genetic material can be DNA or RNA, but never both. So a virus can either contain DNA or it can contain RNA, but viruses never contain both DNA and RNA. Another thing about viruses is they contain no enzymes or very, very few enzymes, but they cannot metabolize on their own. They cannot produce ATP on their own. And so because of this, because of not having a cell membrane and not being able to metabolize, most scientists believe that viruses are not considered to be alive. So not alive. So in general, they are not classified as living things. And you can see that on your microbes to memorize list because the virus names do not come in the genus and species grouping like the other organisms. And also we do not italicize them when we write them. So because viruses do not have their own enzymes, they must use the host cellular enzymes to replicate. So in order to make copies of themselves, they need to use the enzymes found in the host cell to make copies of the virus particles. And because of that, we refer to viruses as obligate intracellular parasites. So they are obligate pathogens, obligate parasites, because they're obligated to use the host cellular enzymes. They cannot replicate on their own. And they're considered intracellular parasites because they must go inside the host in order to use those enzymes. And they are parasites because a parasitic relationship is where one species benefits and the other is harmed. In this case, the virus is going to get the benefit because it's going to replicate, and in doing so, it is going to harm the host cell. So all viruses are obligate intracellular parasites. As I mentioned before, viruses are very, very small, but it's really hard to actually conceive of how small they are. So this figure tries to give a representation of how small those viruses are. So on this side, this represents a human red blood cell, and you've already seen the human red blood cell under the microscope, so you know how small they are. But of course, the human red blood cell is a eukaryotic cell. And remember, eukaryotic cells are very large in the cellular world. So this is just the outer diameter of a human red blood cell. Down at the bottom, you have an E. coli. So this is a bacillus type of prokaryotic cell, and this is the relative size to the human red blood cell of how big an E. coli would be. And then all of these in blue, these are all different types of viruses. And so one of these viruses, the rhinovirus, is a fairly small virus, and rhino means nose. Rhinoviruses cause a lot of the common colds that we have. So if you look at the size of the rhinovirus relative to the eukaryotic cell, you can fit millions of rhinoviruses inside a eukaryotic cell. So these are incredibly small particles, very, very small. And of course, that is why we cannot see them with a light microscope, because just think how small the bacterial cells are. even when we use oil immersion. So the only way scientists really saw them was with electron microscopes, and again, it's been less than 100 years that we've had electron microscopes. When talking about viruses, especially viruses that are involved in disease, it's important to note the host range. So the host range talks about what sort of host cells a virus can infect. And in general, viruses have two types of host range. It can be either a narrow host range or a broad host range. So for a narrow host range, when you're talking about a narrow host range, really you're talking about a virus that can infect only one type of species. And often within that species, only one type of cell. So that would be an extremely narrow host range if a virus can infect only one type of cell in one particular species. And with that, the virus, so this would be a virus. The virus often has molecules on its surface, and these molecules are used to identify receptors on the host cell. So these ligands on the virus are specifically configured so that they will recognize receptors on the host cell. And that's how the virus recognizes its appropriate host cell that it can infect. And this is an example of herpes simplex virus. Herpes simplex virus can cause herpes simplex 1, which is cold sores, herpes simplex 2, which is the STD, and also chickenpox. Chickenpox is actually a herpes virus. And for the herpes virus, the host cell that they are specific for are humans. So herpes viruses only infect humans, and specifically in terms of the cell, they infect leukocytes, which are a type of white blood cell, and neurons. So those are the two types of cells that herpes virus can infect. Another example of a virus that has a very narrow host range is the paramyxovirus. Paramyxovirus can cause a couple different diseases, but one is measles. So this is the virus here, depiction of the virus, and here are the molecules on the surface, the ligands that will recognize molecules on the human cell. So these ligands on the virus will recognize these CD16 molecules on the host cell, and this is found mainly on macrophages. So macrophages are a subtype of white blood cell. And measles is specific for human macrophages. So again, that's a very narrow host range. Conversely, some viruses can have a broad host range. And when you talk about a broad host range, that usually implies that the virus can infect more than one species. And one example of this is rhabdovirus. Rhabdovirus causes rabies. And rabies can infect many different types of mammals. Around this area, the mammal we really think of that can be infected by rabies or is infected with rabies is the bat. Bats actually are the host for lots of different types of viruses. And around this area, we sometimes think of raccoons. Those are probably two of the most common wild hosts for rhabdovirus. Of course, either of these can transmit the virus to domestic pets, to cats and dogs. And of course, all of these can transmit the virus to humans. So humans can also be infected by rabies. So rabies rhabdovirus has a very broad host range. Another virus that has a broad host range is orthomyxovirus, and orthomyxovirus causes influenza. So influenza, and there are many different strains of influenza, and this graph shows you the host range. So influenza A can infect poultry and mammals. And then you have other strains. Influenza B has a more limited host range. It's the seal, pig, and man. Influenza C is very limited. It's just man and pigs. And then you also have influenza D. And with influenza, this causes the flu, of course, and you've heard of bird flu and also swine flu. So sometimes those flus are named from the animals that they come from and infect the human. One of the things I want you to notice is that for all of these strains of influenza, all of them can infect people and pigs. So there's a lot of recombination of the virus between pigs and humans. The last group of viruses that I want to talk about in terms of a broad host range are the coronaviruses. And the coronaviruses have many different species. Many of them produce a sort of common cold type disease, but there are some that produce more severe diseases. Of course, you know COVID-19, which is going on right now, but there's also Middle East Respiratory Syndrome, MERS, and then there is SARS, which is Severe Acute Respiratory Syndrome. Now, all coronaviruses have a broad host range, so of course they infect humans, but they also infect other animals, other mammals. And so for this, SARS was the first one that was discovered. And they think that the natural host is bats. Like I mentioned before, bats are a host, natural host for many diseases. And they think that the virus was transferred to the civet cat and then to humans from there. For MERS, Middle East Respiratory Syndrome, they think it went from the bats to camels. And then for COVID-19, they're not sure how it was transmitted, but they're pretty sure the original host was a bat. And that probably went through some other mammal before it got to humans. So again, coronavirus has a broad host range. Now I want to go through the basic structure of a virus. And the basic structure of a virus is pretty simple. All viruses must have two parts to their structure. The first part is that they have some sort of nucleic acid, genome. So the nucleic acid would be either the DNA or the RNA, and of course this nucleic acid forms their genome, their genes, which determine all the functions of the virus. The shape the virus will have, the host range, and how it will replicate. The second structure that all viruses must have is a capsid. And the capsid is a protein coat that protects the nucleic acid. So again, all viruses must have some sort of nucleic acid and they must have a capsid. There are a couple other structures that some viruses may have, and I will talk about those later. But all viruses must have a nucleic acid and a capsid. All viruses must have some sort of genome, and that viral genome can be either DNA or RNA. Again, it cannot be both. And if you have a DNA virus, that DNA can be in the form of a linear straight line, one long strand, or a circular DNA molecule. And then whether it is linear or circular, it can be single-stranded or double-stranded. So you can have a virus where the genome is composed of DNA, which is linear in long strands, and it could be single-stranded DNA or double-stranded DNA. And you get the same thing with a DNA circular genome. It can be single-stranded or double-stranded. And remember, the double-stranded, that's how we think of DNA, as a double-stranded helix. But in viruses, you can have DNA that is just single-stranded. And with the RNA viruses, you get the same variety. An RNA virus can have either linear chromosomes or circular chromosomes, and those chromosomes can be single-stranded or double-stranded. And for the circular, you just get single-stranded RNA genome. So, but in general, viral genomes, you get a lot of variety because they can be either DNA or RNA, they can be linear or circular, and they can be single-stranded or double-stranded. So again, a lot of variety in the different types of genomes that a virus can have. The important part, though, is knowing whether the virus is a DNA virus or an RNA virus, because this has an effect on how the virus replicates, and it also can have an effect on how it causes disease. So on your microbes to memorize list, I have divided the viruses by DNA viruses and RNA viruses. All viruses must also have a capsid, and the purpose of the capsid is to protect the genome. And for viruses, there are basically 3 different types of capsids they can have. The first type is called a helical capsid, and a helical capsid looks like a tube, basically. And the nucleic acid is inside that tube and protected from the environment. And all capsids are made of capsomeres. The capsomeres are these little repeated structures that make up the capsid. And again, these are all proteins. So the capsomeres are similar proteins that bind together to form the helical capsid, the capsid. Second type of capsid is polyhedral. Polyhedral means many sides. So these capsids look like those many-sided dice, more than 6 sides. So they're very geometric looking with all these faces and very regular. And again, they are composed of capsomeres. So each one of these little circles here represents a capsomere. So they have the same structure and they bind together to form the polyhedral capsid. And again, the nucleic acid is protected inside. Also on the polyhedral capsid, you will notice there are these glycoproteins, these protein fibers that are extending off from every corner. And those will be used as ligands on the virus to recognize and attach to the appropriate host cell. The last type of capsid a virus can have is a complex capsid. So this is a complex capsid, and if you look at the structure, it is complex. Often with this, you have a polyhedral head and a helical sheath, and then you have these tail fibers here. And the tail fibers are what complex capsids use to identify and bind to the appropriate host cell. So this is a complex capsid, and you find these mainly on bacteriophages. And bacteriophages are viruses that infect bacteria. While all viruses must have a capsid and a nucleic acid, some animal viruses, and when I say animal viruses, I mean viruses that infect animal cells, some animal viruses may have an envelope. And an envelope is a phospholipid bilayer that surrounds the capsid and the nucleic acid. So the envelope is exterior to the capsid and the nucleic acid. And also embedded in the envelope are these protein spikes. So these would be the spikes that are the ligands that interact with the host cell, identifying and binding to the appropriate host cell. And influenza is an envelope virus, as is coronavirus. And coronavirus is actually called coronavirus because it looked like a crown was sticking out on the virus. So, and those were the protein spikes that are embedded in the envelope. When studying microbes, it's very important to be able to culture them in the lab, to grow them in the lab. Because by being able to grow the microbe in the lab, scientists can study it better to determine its function, its structure, and also how to cure diseases that are caused by that particular microbe. So culturing viruses is also very important to do in order to understand them and the diseases they cause. Now viruses, if you remember, viruses don't have any enzymes, they don't metabolize on their own, so they can't replicate on their own. The only way they can copy themselves is by infecting a host, going inside a host cell, and then they can use those enzymes, the machinery of the host cell, to make more copies of themselves. So in order to culture the viruses in the lab, you have to be able to culture the host cells first. Culturing bacteriophages is relatively easy. Remember, bacteriophages, these are the viruses that infect bacteria. And in order to do this, the first thing you have to do is be able to culture the bacteria. So often this is done on a plate, a TSA plate, and you create a lawn of bacteria over the plate. That means you spread the bacteria evenly all over the plate. And then once the bacteria has grown, then you introduce the phage particles. So you add the phage particles, and wherever the phage particles are infecting the bacteria, they will lyse the bacteria and you will create these empty spots. And so that's evidence that the phage has infected the bacteria and lysed the bacteria. So in these empty spots, you have a lot of phage particles that have been generated. So if you swab these empty spots and transfer that swab to another lawn of bacteria, then you will be transmitting the phage to that other sample of bacteria. Culturing animal viruses can be a little bit trickier. So remember, viruses again cannot metabolize on their own. They can't replicate on their own, so you can't take the viruses, put them on agar, and have them grow and replicate. So for the animal viruses, you need to be able to culture the host cells first. And this can be difficult when you're culturing cells from a multicellular organism like humans. Hepatinovirus is a virus that causes hepatitis, and it affects the human liver. So hepatinovirus has a very narrow host range. It only infects human liver cells. So if you want to grow hepatinovirus in the lab, you need a source of human liver cells, which is kind of difficult to get. But if you do actually get some human liver cells and put them into culture in the lab, the thing about the cells from an actual organ is that they will only last about 2 weeks when you are culturing them in flasks or in petri dishes. Even if you give them all the nutrients they need, they will only grow for 2 weeks. And so that's not enough time to get significant amounts of hepatinovirus to really study. So what scientists have done instead is, instead of culturing just pure liver cells, they culture or they grow cancerous liver cells. So they have found tumors from livers and cancer. The basic definition of cancer is a cell that is going through cell division uncontrollably. So it's uncontrolled cell growth. And that's what cancer cells do. They have a problem in the cell cycle regulation, and they're just dividing continuously. So that's actually good for culturing viruses, because these types of cells will grow indefinitely, basically forever in culture. So to often culture viruses, we use transformed cells, which are cancerous cells from the host cell that the virus infects. Orthomyxovirus, I have mentioned that before, that includes influenza. And actually, ortho refers to ornithology, which is the study of birds. So remember, this has a very broad host range, so we don't have to rely just on human cells. And actually, the virus for influenza, when we are producing it for a vaccine, we actually grow it in eggs, in poultry eggs. So they take a fertilized egg and they can inject it with a virus at different places to grow the virus so that there is enough virus to use for the vaccine. And that still is currently how they generate virus for the vaccine, is by infecting fertilized poultry eggs. All viruses go through a basic life cycle, and it's important for you to know the basic stages of the life cycle, and then for the different types of viruses to know the specific life cycles. So for this, I want to go through the basic 5 stages, which are attachment, penetration, biosynthesis, maturation, and release. And basically what is happening at each stage is that during attachment, that is where the virus is identifying and attaching to the appropriate host cell. Then in penetration, The virus is getting its genome into the host cell. So it is getting its genome into the host cell, and often what happens is it is degrading or breaking down the host genome or the host DNA. Once that has been accomplished, then the virus can take over the machinery of the host cell and go through biosynthesis. Biosynthesis is where the virus is synthesizing copies of its genome and its capsomeres. And remember, the capsomeres are the building blocks of the capsid. So once these two components have been synthesized, copies of the genome and the capsomeres, then the virus goes through maturation, and that is where it assembles those together and forms mature virus particles. Once all of those mature virus particles have been assembled, then the last stage is release, and that is where the virus is released into the environment. And then this life cycle starts all over with all those offspring viruses identifying and attaching to appropriate host cells. So this is basically what goes on in a generalized viral life cycle. And as I go through the different bacteriophages and animal viruses, you will have to know their specific life cycles. Now I want to go through in more detail bacteriophages. And bacteriophages are viruses that infect bacteria. And the name is a little unusual. Bacterio refers to bacteria, and phage means to eat, because bacteriophages were identified before we had electron microscopes. So before scientists had the ability to actually view the viruses. So what happened is you had scientists who were studying bacteria, and they would grow lawns of bacteria. And once in a while, they would notice that in that lawn of bacteria, they would get these clear spots where all the bacteria had disappeared. And when they looked at those and saw that all the bacteria were gone, they thought that something was eating the bacteria, because how else would the bacteria disappear? So what they also noticed, and what I mentioned before, is if they swabbed this area and put it onto another plate, whatever that was eating the bacteria would be transferred to that plate. So even though they couldn't see it, they knew something was killing the bacteria, so they called it a bacteriophage. So something was eating the bacteria. Now we know, of course, viruses don't eat bacteria, they're lysing the bacteria. So they were killing the bacteria in those areas, but that's where the name comes from. All bacteriophages have exactly the same structure. So all bacteriophages have complex capsids. And I mentioned this before when I was talking about the different types of capsids. They all have a polyhedral head and a helical sheath, and then tail fibers, all of these tail fibers. So that is the type of capsid they have. And in terms of the genome, all bacteriophages have a DNA genome that is circular and double-stranded. So all bacteriophages have this complex capsid, and their genome is composed of DNA, which is circular and double-stranded. There are two different types of life cycles that bacteriophages can have. They can have either the lytic life cycle or the lysogenic life cycle. This is a diagram of the lytic life cycle of a bacteriophage. And as you can see, it goes through the 5 stages very directly. So going through in more detail the different stages. First stage is attachment. Attachment, remember, is where the bacteriophage is identifying and binding to the appropriate host cell. So to do this, the bacteriophage uses those tail fibers. So it's going to use the tail fibers to recognize and adhere to the appropriate host cell. Once it does that, then it moves to penetration. So penetration is getting the DNA from the capsid into the cell. And bacteriophages do this by injection. So what they do is that helical sheath contracts, gets shorter. So here the helical sheath is contracting, and it injects, kind of like a hypodermic needle, the DNA into the host cell. So it injects it into the host cell. And when doing this, the capsid remains on the outside of the bacterial cell. So it's only DNA that is going in. So also during penetration, the phage is injecting its DNA into the host cell, and then the host chromosome gets degraded. It gets broken down into pieces because the phage DNA is going to take over control of the host cell. So once that has been accomplished, then it goes through biosynthesis. Biosynthesis is where you're making all the different components for the virus. You're copying the phage DNA, the genome, and you are also making all the capsomeres and the tail fibers and other protein components. And this is all happening in the cytoplasm. So in the inside of the cell, cytoplasm. All right, once all the components have been made, all the components are made, then you move to maturation, and maturation is where those components are assembled into mature viral particles or virons. So now you get assembly of all those components into the mature virons, and once they have all matured, they've all been assembled, then you move to release, and lytic phages release by lysis. So they rupture the cell, and all these phage particles are released, and the cell, of course, cannot survive lysine, and so the cell dies. The other type of life cycle is the lysogenic life cycle, and the lysogenic cycle is very similar to the lytic life cycle, but there's one important difference. This is a diagram that goes over the lysogenic life cycle of a bacteriophage. And you can see that there are sort of two parts to it, a lytic cycle and a lysogenic cycle. So first, just to get you used to this diagram, I'm going to go through the lytic cycle. So this, number one, the phage attaches to the host cell. So it went through attachment, and it looks like it is going through penetration, injecting the DNA into the host cell. So after penetration, you get to a point where you have the phage DNA and the host chromosome. Now, if this was a lytic life cycle, that host chromosome would be degraded, and then you would go through biosynthesis and maturation. So this looks like it is at maturation right now, where you have the mature viral particles, and then the last stage is release by lysis. So it released by lysis. So that is basically the lytic cycle that I talked about before. A lysogenic life cycle is very similar. It would start the same way with attachment by the tail fibers, penetration by injection, and then you would get to this point where you have the phage DNA and you would have the host chromosome here. Now this is where the difference is. Instead of breaking down the host chromosome and taking over the cell and going on with the cycle, instead a lysogenic phage will take its chromosome and splice it into the host cell's chromosome. So instead of breaking down the host cell's chromosome, the phage DNA splices into the host cell's DNA. And that creates what we call a prophage. So when you have that phage DNA spliced into the host cell chromosome, that is called a prophage. Pro before phage, before making the phage particles. And when this happens, the bacterial cell has no idea anything happened. The bacterial cell can still do its regular thing. If it has flagella, it can swim around, it can get nutrients, it can metabolize, it can respond to stimuli, and it can replicate. And every time that bacterial cell divides, all of its daughter cells will have the prophage in it. And so this can go on for the life of the bacterial cell. Nothing may ever adversely happen to that bacterial cell. But if these cells that have a prophage in their chromosome, if they are exposed to a trigger, and with bacteriophages, often that trigger is a change in temperature. So if they're exposed to that trigger, which again often is a change in temperature, then that prophage will splice out. So if they're exposed to this trigger, the prophage will splice out, and then you get back to the situation where you have a separate phage DNA and separate host chromosome. And once that prophage has spliced out, then you continue in this cycle. So then you go through biosynthesis, maturation, you get those mature virus particles, and once they have all assembled into mature virus particles, then the virus will release, the phage will release by lysis. And again, that will kill the host cell ultimately. So this is the lysogenic cycle of a bacteriophage. When going through the phage life cycles, I think sometimes it's good to create like a chart to compare and contrast. So on one column, the first column would be the stages: attachment, penetration, biosynthesis, maturation, and release. And then you can make two other columns, one for the lytic phage and the other for the lysogenic phage. So for this, the lytic phage, it's going to use the tail fibers. For attachment, it's going to penetrate by injection. Biosynthesis will occur in the cytoplasm. Maturation will occur in the cytoplasm, and releases by lysis. And the lytic phage is going to go straight through this process, and usually from attachment to release, it takes about 20 minutes. Lysogenic phage basically does the same thing. So you have attachment is by the tail fibers, penetration is by injection, but then during penetration, you're going to get formation of that prophage. So the formation of that prophage, which splices, the phage DNA splices into the chromosome, and then that bacterium can still continue doing its normal thing until you get a trigger. If you get that trigger, which for prophages or for lysogenic phages, it's a change in temperature, then it goes back into the cycle where biosynthesis is going to occur in the cytoplasm, maturation in the cytoplasm, and release by lysis. So the way I kind of think about the lysogenic life cycle is it is basically like the lytic life cycle, except at the stage of penetration, that forming of the prophage is like hitting pause. So it's kind of like watching a movie. A lytic phage is if you watch a movie all the way through, you don't have any interruptions. A lysogenic life cycle is like you start the movie and then you hit pause for a moment. And then once you have the trigger to go back to the movie, you hit play again and you go through it again. An important process that relates to phages is called transduction. And transduction is when bacterial genes, so bacterial genes are transmitted from one bacterium to another by a phage. And we will talk about transduction again when we get to genetics, microbial genetics. But transduction is basically defined as when bacterial genes are transmitted from one bacterium to another by a phage. So the important parts of this is we're referring to bacterial genes, prokaryotic genes, and the phage is moving them from one bacterium to another. And there are two types of transduction: generalized transduction and specialized transduction. Generalized transduction can happen with any type of phage infection, so lytic or lysogenic. And this figure goes over the basic process. So this shows the life cycle of a phage, so starting with attachment and penetration, and then biosynthesis and maturation, and release. Now what basically happens is you have the first bacterium. So this is the first bacterium that is infected with a phage. So again, the phage goes through attachment, it's going through injection, and then the host chromosome is degraded. So the host chromosome is cut up into little pieces so that the phage DNA can take over. Well, during maturation, so it goes through biosynthesis and maturation. During maturation, when you are assembling the mature viral particles, accidentally, the phage, one of the capsids, packages the DNA from the host cell. So one of these pieces of DNA is accidentally packaged into a capsid. So this particular capsid, instead of phage DNA, it contains bacterial DNA. So when this cell finishes out the phage life cycle, maturation, and release by lysis, it's going to release all of these phage particles, and then this phage will be able to use the tail fibers to recognize and adhere to another host cell, and it will penetrate, inject the DNA, but it is injecting bacterial DNA. So remember, it's taking that DNA that was accidentally packaged into this phage capsid, and it's injecting that DNA into the second bacterial cell. So this is a second bacterial cell. And then that DNA can splice into the second bacterial cell's chromosome, and now this bacterial cell has acquired a new gene. So it's acquired some sort of new gene, which will produce a protein product and give that cell a new characteristic. The second type of transduction is called specialized transduction, and this is associated only with lysogenic phages, because you need the formation of a prophage. And for this, you go through the same phage life cycle, and again, it's a lysogenic phage, so there is the formation of a prophage. So you have this pink prophage DNA, or phage DNA, spliced into the bacterial DNA. And then when this cell gets the trigger, the phage DNA splices out, and when it does so, it takes with it a small piece of the bacterial DNA. So in this case, it's the gal gene, which I'll talk about in a second. So the gal gene was part of the bacterial DNA, and when the prophage splices out, it takes that gal gene with it. So then as the phage replicates, as it copies its DNA, all the phage particles, all of the mature phage will contain that bacterial gene with it. And so when these phage particles infect the next type of bacteria, they're going to inject the phage DNA, which contains that bacterial gene, and that DNA will splice into the chromosome, forming the prophage and also containing that gal gene. So whatever protein that gene produces, this bacterium now has that ability. And the gal gene is actually a gene that encodes an enzyme that can utilize galactose. So this original cell was able to produce an enzyme that breaks down galactose. The second cell was unable to break down galactose, but because they had the phage infection and acquired that gene, now the cell now has the ability to break down galactose. So it has been transduced. A bacterial gene was transferred from one bacterium to another by a phage. So that's the definition of transduction. Phage conversion is the term we use when a bacteria is given a new ability from being infected with a phage. An example can be with that specialized transduction with the galactose gene, the gal gene. And so again, remember that the original bacterium had the gal gene, and then when the prophage spliced out, it took that gal gene with it. And so every phage particle now has a copy of the gal gene. So when that phage infects a new bacterium that did not have the ability to break down galactose, it could not utilize the galactose because of that gal gene that went along with the phage DNA. It now has that ability to metabolize the galactose. And you can actually see that there's media that contains compounds that if bacteria are able to utilize galactose, they turn blue. So this is an example of a plate with transduced bacteria on it. So the original bacteria were were unable to use galactose, and when they grow on the media, they are the normal white color. But those that have been transduced, that were infected with the phage, and they acquired the ability to produce an enzyme to break down galactose, they turn blue when grown on this media. So this is an example where you can actually see the different types of bacteria that have been transduced by the phage.